This is our presentation on the phylogeny of the Carcharodontis genus across the globe. My name is Eric.
And I'm Sarah.
So this is just kind of an overview of what we're going to talk about throughout our presentation. So for our research, we decided to choose the genus Carcharodontis, which is a shark genus in the family Carcharodontidae. They're also known as the requiem sharks. So there are 12 genera in this family, the Carcharodontidae, and this genus is the largest of the 12. It has over half the species in the entire family in this one genus. Also, there are 35 extant, meaning they're still alive species, and 8 extinct species, but it is very likely that more will be found. So here's just a quick overview of a few of the different species that are in this genus. We chose 6 just to show you real fast, but we did have 15 overall for our research. We tried to choose 1 from each geographical location, but if you take a look, usually about the dusky shark, the nervous shark, and the black-nose shark all have a similar caudal fin. The white creek, the silvertip, and the creek whaler all have a similar fin and caudal fin in the back. It's more barbed. And just taking a look at the general colors of all the sharks, the white creek shark and the dusky shark are the outliers. The dusky shark is more of a maroon or red. The white creek is more of a greenish, while the rest are gray or green, gray or tan. So like I said before, we did have 15 sharks that we used for our research. And we kind of split them into 4 geographical locations. While they are not perfect separations, they kind of give you a good overview of where they're most likely to be found and who they're nearby. So for this first region, we have 6 sharks, more so near the Indian Ocean on the coastlines of South Asia, and especially above the coastline on the upper coastline of Australia. In our second geographical location, this is our Pacific Ocean region. This one does have the most overlap with the Indian Ocean and, like I said before, the coastline of Australia. But this one is also found, quote unquote, on the other side of the Pacific Ocean, more so nearby the Americas. Our third geographical location only has 1 shark, but they are found on the Atlantic side of the Americas, so on the coastline of Central and Southern America. And then for our last region, kind of is a wide region, but it covers the entire global. So these 5 sharks are found pretty much everywhere. There's a pretty large mix of them all across in all the oceans. So the next question is the genus Carcharodontis monophyletic? So in our literature that we used for pretty much our entire presentation, it kind of compared many different studies and kind of the evolution of a study of a specific gene. So according to it, originally, the family Carcharodontidae was found to be paraphyletic, but the genus Carcharodontis was originally monophyletic. And that was proven due to Allozyme's data analysis. And Allozyme is basically just a different variant form of an enzyme, which differs based off of the allele that you have. And so originally they used the blue shark as an outgroup for a group of Carcharodontis species. But it was actually found that the Prunus genus, which is the blue shark, it actually has a pretty close relationship with the Carcharodontis, almost very identical. And so in a new study that they used, there was a few studies before this, but they took 17 species of the Carcharodontis sharks and 4 closely related species, and this resulted in a final paraphyletic tree, 2 species from other genera and 1 species from another family. If you take a look all the way to the left, you can see that in pink, the Carcharodontis genus is highlighted. They're mostly bundled together, but there are a few exceptions.
So this is showing phylogenies from a study by Manet, Dosset, Akbulut, showing 7 species from the Carcharodontis genus, as well as 3 outgroups, which are highlighted in pink. So to the left, there is the maximum parsimony analysis of ribosomal ITS12, which is more similar to what we did. And then on the right is Kimura analysis of the same gene. So looking at the bootstrap values, you can see there is a very high probability of these relationships being accurate. And this shows that despite there being physical differences between these sharks, there are not as many genetic differences. So here we have Carcharodontis isodon and Carcharodontis porosus. So you can see their dorsal fins have some differences in their shape. Same with their pectoral fins. On the left, it's much longer, and then with their anal fins. Despite this, though, they are actually very closely related and right next to each other on the phylogenetic tree. They are one of the most closely related in the phylogenetic tree. So for our phylogenetic tree construction, we used 15 species from the Carcharhinus genus, and our outgroup was Hemigalius microstoma. So the outgroup is in the same order as our genus, so we thought that would be a good choice. And for our gene, we used NADH dehydrogenase subunit 2. So this is a gene that's involved in electron transport in cell respiration in the mitochondria, and it was present in all the genes of all the species of our genus as well as our outgroup. So here's our phylogenetic tree. The ones labeled red are sharks found in the Indian Ocean. As you can see, there's some groups, but then also some outliers. And then those in blue are sharks from the Pacific Ocean. Again, a little group and an outlier. And then we only had one shark from Central and South America, highlighted here in purple. And although it's the only one of that area, you can see how it relates to sharks from seemingly very different locations. And then we have our sharks labeled in green, which are those that can be found globally. And again, we have a close group and then an outlier. So comparing our tree to the studies tree, we include location while the study does not. We also, despite having different species, do show similar relationships. So both our tree and the studies tree show the relationship between C. falciformis and C. agronotus. So those two species are rather distantly related and have a not very recent common ancestor. We also use different genes. So we used NADH dehydrogenase and they used ITS1-2.
So taking a look at our overall macroevolutionary question, this one is more so geographical in terms of relating to phylogeny is what kind of the approach that we took, but more so in the text or the literature that we were using, they were more so taking a look at the overall just DNA phylogeny relationship. So our overall question was, what is the relationship and history of the species from the genus Carcharhinus as they evolved and changed through through environmental differences and speciation? So if you take a look at the two quotes that we found from our literature, it was found that one species was more closely related with two others based off of just morphological structures, so similar tooth shape and vertebral characteristics. But it was found that this is actually not supported by the Allozyme data analysis, because even though they have similar behavior and body shape, color pattern, and tooth shape, that's more so a factor of convergent evolution rather than relatedness. So kind of what Sarah was talking about earlier, where two sharks that looked very different actually were pretty closely related and are one of the most closely related. Taking an opposite kind of viewpoint of that, like two sharks who may look extremely very similar aren't necessarily more closely related just because they look the same.
So in conclusion, we have found that while sharks of similar geographic locations were more likely to be closely relations, there are some exceptions to that. So this shows that there is definitely some convergent evolution happening in Carcharhinus phylogeny.